Thank you, Rick. Good afternoon, everybody. Yes, so my talk's going to explain to you some of the data from our laboratory, how I hope to convey to you how prion diseases appear to hijack the gut-associated lymphoid tissue, such as the Peyer's patches, to make their way from the gut lumen to the brain, where they eventually cause disease. But as will become apparent during this talk, it fits very nicely with the previous two talks on parasite infections as well, so it's a useful session to begin. Close this down. Okay, so the disease we work on in the group is known as the TSEs, or transmissible spongiform encephalopathies. These are essentially infectious diseases of the brain which cause a sponge-like pathology within the brain. I should point out these are not encephalitis. So although I consider myself an immunologist, these diseases don't cause a typical or classical inflammation within the brain, so they shouldn't really be confused with other neurodegenerative diseases, such as multiple sclerosis, which will cause a classical T-cell-mediated inflammation in the brain. And of course, with mouse EAE models as well, they're quite distinct diseases. But these are invariably fatal neurodegenerative diseases. Some of these diseases we think have some genetic association and appear to occur sporadically, but there are also infectious forms of these diseases as well. And I've summarized in this title some of the key diseases within this group, and we have some diseases which we find in humans, such as Kuru. This is essentially a historic disease, was originally identified in the Fore people in Papua New Guinea, and this disease was very important because it helped to show that these diseases could be transmitted, because the scientists that originally discovered this disease realized that this disease was being spread by cannibalistic rituals. So he went there, told them to stop eating each other. Fortunately, they didn't eat him, and the disease is essentially historic now and appears to have died out. I'm going to come back to this one in a moment. We have disease scrapie, which we find in sheep and goats. BSE, bovine spongiform encephalopathy, which I'm sure you're all very familiar with. And of course, this disease appears to have been spread to humans as well, certainly through infectious food, causing variant CJD. Probably one of the most interesting ones is chronic wasting disease. We find this in deer and elk and moose and wapiti, particularly in North America. I consider this interesting because it's the only prion disease I'm aware of that has been identified in free-ranging animals. So you find this in both free-ranging deer and elk or cervids, but also in farmed species as well. And of course, important for a mouse immunologist, we can transmit these diseases reasonably readily to mice. So we have a very tractable mouse scraping model which we can use in the laboratory. These diseases are typically characterized by their neuropathology which they cause. So on the left-hand side, we can see a typical H&E stain from a nice healthy mouse hippocampus. And this contrasts with that from a mouse with clinical disease, which you probably can't see, but there are many small vacuolations in here, typical of the neuropathology the disease causes. And this is coincident with the strong accumulation of an abnormally folded version of a host protein within the brain. This is the prion protein. And you see, this is a normal mouse, and in an infected mouse, we see some heavy deposits of this abnormally folded version of the normal cellular form of the prion protein. And we call this the PRPC, or PRP scrapie form. And we consider these diseases to essentially be infectious proteins, although the jury is still out as to whether this is indeed the case. There are still some aspects of this hypothesis that we're uncertain about. But essentially, the PRPC, or the cellular form, is expressed on almost all cells within the mammalian body. And through some unknown mechanism or voodoo, this becomes abnormally folded into this PRP scrapie form, and this confers some very distinct biological and physical-chemical characteristics. This now becomes infectious, so by this unknown mechanism can auto-convert more PRPC into the infectious form. This is also remarkably stable as well. It's very difficult to destroy this protein. As an immunologist, what we find is if you peripherally expose an animal to prions, we find
substantial accumulation in the lymphoid tissues of those infected animals before we see any accumulation within the brain. And this is important because if you were to remove the spleen from this mouse before you expose it to prions, it takes much longer for the infection to actually spread into the CNS, suggesting that actually accumulation within these lymphoid tissues is important for the spreading of the disease to the brain. We've rephrased this neuroinvasion, so the spreading from the site of infection to the brain. And if we look within the lymphoid tissues and stain for the prion protein, we can see a very specific site of accumulation. We see accumulation of the prions upon a stromal cell known as the follicular dendritic cell. These aren't related to macrophages or those classical dendritic cells. These are a stromal cell within the B cell follicles. And from data I don't have time to explain today, but we're reasonably confident that these cells are actually replicating the prions. And so my group has been particularly interested in how are prions exploiting the lymphoid tissues to make their way into the CNS. And we're focusing on oral transmission because one of the major routes of transmission for some of the acquired prion diseases appears to be oral exposure. And we could use a mouse model. So what we do is we take this is not an earplug, it's a mouse food pellet. We take a brain homogenate from a prion disease affected mouse. We make an homogenate, apply a small blob onto a mouse food pellet, let it dry, then allow the mouse to eat that naturally. So we put it into a cage without any bedding, just put the pellet in there, leave it overnight. If it's ate all its pellets, it can go back and play with its friends. And then about 340 days later, in our control mouse, this will eventually develop clinical disease. And these characteristics absolutely astonished me because I used to work on parasites, and I worked on T. brucei, and the parasitemias after the first few days would go crazy. But even though we're using a natural route of just eating, it's not gavage, we're using a reasonably low dose, most of our mice will succumb to disease within a week of each other, which I still find quite remarkable. But it's very useful for designing experiments. Okay, so we know that after oral exposure, the agent has to accumulate or replicate within the gut-associated lymphoid tissues in the intestine. So, for example, this is a whole mount from David and Anuj in the group of a pest patch in a mouse intestine. And we know that if we take a mouse which doesn't have any pest patches, feed it scrapie, that mouse will not develop clinical disease. So the agent has to replicate within the gut-associated lymphoid tissues to spread to the nervous system. What you can see here, these finger-like projections are the villi, and this is a pest patch. And these are we see here is a specialized epithelium which overlies, we call this a follicle-associated epithelium, it overlies the B cell follicles within those pest patches. You can see there's about six follicles within this structure. And what we've been interested in the lab is how do the prions make their way into these structures, and how do they manage to make their way to those follicular dendritic cells where they replicate. And Judy had her big three. I've summarized in this slide my big three, and we have the M cells, the dendritic cells, and the follicular dendritic cells. And work over the many years in my group has been trying to dissect the cells and molecules within the gut-associated lymphoid tissues, which are important for, or whether they are, of course, whether they are important for conveying prions from the gut lumen down to these follicular dendritic cells from where they appear to then spread into the nervous system and cause disease. And so I'm just going to show you a couple of examples of some of the kind of experiments we've done in the lab to try and dissect this cellular route. So the first question we want asking in this particular talk is whether M cells are the route through which prions can make their way across that gut epithelium. If I actually... no, it's okay, I won't go back, I'm using a computer. Here again we have a nice whole mount looking down onto those epithelia overlying those B cell follicles within the pest patch. And the green cells here are stained with an antibody against GP2, which specifically picks out the M cells. And M cells are a specialized epithelial cell within this epithelium here, and they're specialized for transcytosing antigens or macromolecules across the gut epithelium, so they can then be sampled by macrophages, dendritic cells, and lymphocytes, which can then mount an immune response if needs be. So we wondered,
because we know that pathogens such as salmonella can exploit these cells to make their way into the body and cause the disease, we wondered whether prions may also exploit these cells to make their way into the body. And the way we were able to get at this was by exploiting the factors which are important for maintaining M cells in their differentiated state. So this dotted line shows where the M cells would be in the gut epithelium. This is the gut lumen, this is the epithelium, and immediately beneath the epithelium is a network of stromal cells expressing this chemokine, which we've stained in green here, called rank ligand. And this has been shown to be critically important for maintaining M cells. And so if you block rank ligand signal, for example, by injecting anti-rank ligand, within a few days, you completely block the formation of M cells. We've got a nice model here where we can expose our mice to prions and see whether, if we take those M cells away, can the prions cross the gut epithelium and will the mice get sick? And the answer would be no, the prions can't get across, and no, the mice don't get sick. And we can see that here. In our control mice, we've stained here, this brown stain is picking out the prion accumulation on those follicular dendritic cells within the B cell follicles. And we can see a nice heavy accumulation within about 10 weeks of oral exposure. And in the mice in which we blocked the rank ligand signal and got rid of those M cells, we can see we've blocked the accumulation of prions in those pairs patches throughout the intestine. And then if we follow the remaining mice, see whether they go on to develop clinical disease, we can see our control mice, again, around 340 days after exposure, they all succumb to clinical prion disease, whereas those which didn't have any M cells, they were all free of disease a year and a half after exposure. And we looked throughout the bodies of these mice, and we couldn't find any signs of prion disease. So we think M cells are an important route through which the prions initially cross the gut epithelium. So of course, once the prions have crossed the epithelium by M cells, we still want to know how they then make their way from the lamina propria down to those follicular dendritic cells where they then replicate. Do they just, knock about a bit like in a pinball machine, eventually making their way down to the follicular dendritic cells, or is there some kind of cell-mediated process through which they're carried? And this is just a whole mount graph from the MAC blue mice, which we have at the Roslin Institute. And this just shows that immediately beneath the epithelium, we can see quite a dense network of macrophages or dendritic cells beneath that epithelium. And so we wondered whether, after their transcytosis by M cells, are they then picked up by a macrophage or a dendritic cell and then carried down towards those follicular dendritic cells. And so we originally worked on this about, embarrassingly, 7 years ago now, work that Claudine did, using the CD11C DTR transgenic mice, in which the diphtheria toxin receptor is expressed on CD11C, which is expressed on macrophages and DCs in the gut. And you can see if you treat mice with these mice with diphtheria toxin, you ablate all the CD11C positive cells in the pairs patch. And importantly, though, when you feed these mice scrapie, whereas the controls again accumulate prions on their follicular dendritic cells and go on to develop clinical prion disease, these mice in which these cells were ablated at the time of oral exposure, there was no prion accumulation on these follicular dendritic cells, and importantly, they again didn't go on to develop clinical disease, suggesting that once the prions have been transcytosed by the M cells, they're perhaps then picked up by dendritic cells and may be carried down towards that. Of course, this data doesn't prove that they are carried down towards it. It just shows that when we take those out, they can't get their way down there. And so, I'm using the wrong computer. Sorry. In the next slide, this is work that Barry Bradford's been doing in the laboratory. We then created a compound transgenic mice to test the hypothesis that these cells, these macrophages or DCs up here, are indeed carrying the prions down to the follicular dendritic cells, which are here. And so to do that, we manipulated some of the chemokine receptors expressed on the cells up here. And we know that follicular dendritic cells express CXCL13. It's a chemokine. It used to be known as B lymphocyte chemokine. And it's important for pulling cells into the B cell follicles. And it attracts CXCR5 chemokine receptor expressing cells, such as B cells, follicular T helper cells, and dendritic cells into this B cell follicle. So by using Krielox, we created mice in which the CXCR5 receptor was specifically ablated only on CD11C positive cells. And so these cells would not be able to migrate down towards the B cell follicle. Now, it was important to go to this
very painful method of doing this, because if we were just to take a CXCR5 knockout mice, these mice lack pairs patches, and I showed in one of my very first slides that if we don't have any pairs patches, the mice don't get sick. So yes, the dendritic cells won't be able to move down to B cell follicles, but there aren't any pairs patches, so the mouse won't get sick anyway, so it's pointless using that model. So we had to create this transgenic mouse. And hopefully you can see the black staining here depicts the prions. So you can see in our control mice, we've got typical prion accumulation on those follicular dendritic cells, and it's blocked, or at least the early accumulation is blocked in the mice when these cells can't move down here. We think we can see some accumulation here, so it appears that these cells are probably retained in this apex and subepithelial dome as we expect here. Now, the talk isn't entirely just restricted to prions. Barry's also exposed these mice to Trichuris muris, which was nicely introduced by Judy and in the previous talks as well. And we can see that whereas in our black 6 mice, when we expose these mice orally to T. muris, within a few weeks after exposure, they expel most of the worms as we would expect. Whereas these mice are unable to clear the disease, and their large intestines are packed full of worms. So Barry's keen to follow up these data. Okay. Right. So our next big 3 is location, location, location. So I've described some of the cells within lymphoid tissues which we think are important for conveying prions into those gut-associated lymphoid tissues. But what none of these experiments answer is which are the important gut-associated lymphoid tissues. As mouse immunologists, and we think we often study gut infections, we often think of pairs patches being the bee's knees. And we think I think location is important. And if we look at the patch structures throughout the intestine, we can see there are pairs patches in the small intestine, but there are also equivalent structures within the large intestine. And these have, in the pairs patches, they've got follicle-associated epithelia, as they have down here. So there are M cells down here. There are also B cell follicles in these structures as well. And within those structures, there are also follicular dendritic cells. So each of these structures has the potential to accumulate and replicate prions, and it may be that the prions are actually going into these structures and then spreading from there into the nervous system, and that this is just a secondary effect due to dissemination, etc. But important for the subsequent slides is these structures develop in utero, as Cecile mentioned, via lymphotoxin-dependent stimulation. But there are also many other smaller solitary lymphoid follicles throughout the intestine, which are termed isolated lymphoid follicles. These are essentially, I call them like mini pairs patches. They're just individual B cell follicles, we can see here. They can be either these immature forms, where they're just small clusters of B cells, or what we've termed essentially mature isolated lymphoid follicles. And the red staining here is representing the follicular dendritic cell network. So, like I say, essentially a mini pairs patch. We know that there are M cells in the epithelia overlying these structures as well. And you can find these throughout the intestine as well. These are some whole mounts that David's done. You can find them in the small intestine, in the cecum, and in the large intestine. But these develop postnatally. They still are dependent on lymphotoxin, and they're also modulated by environmental factors such as the microbiota. The microbiota can stimulate or inhibit the development of these structures in particular sites. Now, we're interested in the location because it's an interesting scientific question, but also it has some important applications as well. That being that if we look at the large intestine associated gut-associated lymphoid tissues of naturally prion disease affected hosts, we can find prion accumulation there. This is showing, I think it's from a scrapie affected sheep, but the same has been found in deer as well. If we take the ramels, so the rectal associated lymphoid tissues, at a point during the infection, we can see accumulation of prions within those structures. Likewise, if we look at the there have been retrospective analyses of appendixes from humans in an attempt to try and gauge the potential incidence of variants usually in the human population in the UK. And a small number, but certainly some appendices have been identified in which prion accumulation, or at least staining for prions, appears to have been identified. And that would suggest that there's a potential incidence of 1 in 2,000 people in the UK may have or maybe accumulating at least anyway, variant CGD prions in lymphoid tissues. So it's an important thing to know what is the involvement of these structures. And so to do so, David created a mouse model in which the mouse, at the time of oral exposure, only had FTC-containing gut-associated lymphoid tissues in the large intestine at the time of exposure, not in the small intestine. And we did that by manipulating those isolated lymphoid follicles, because what we can do is we can treat a pregnant mouse with this drug here, lymphotoxin beta receptor Ig. It blocks the formation of Peyer's patches during in utero in the pups, but because the isolated lymphoid follicles form postnatally, it induces the formation of large numbers of FDC-containing large associated large intestine isolated lymphoid follicles in the large intestine within 21 days after exposure. We don't see them in the small intestine at this time after birth. Whereas if we wait until 56 days after birth, we find these structures throughout the intestine. So we can use this to specifically look at the involvement of these structures and these structures in oral scraping pathogenesis by studying the absence of these ones. And I'll go into this slide first. So what we've done here is to use that model and take the mice at 21 days old, orally expose them to prions, then take their intestines about 10 to 15 weeks after oral exposure and look for the accumulation of prions. And you can see in our control mice, as we would expect, we see heavy accumulation of prions in the Peyer's patches of these mice. But interestingly, if we look at the cecal patches and the colonic patches, we don't see any accumulation of prions in our control mice. These mice don't have any FDC-containing ILFs throughout their intestines. These are the control mice at the time of oral exposure, whereas these mice only had FDC-containing ILFs at the time of oral exposure, and we still don't see any accumulation of prions in their lymphoid tissues. And importantly, those mice didn't go on to develop clinical disease, whereas the control mice, about 380 days in this particular case, they'll develop clinical disease. As we'd expect, only 9 of those mice did, suggesting that the large intestine is not an important site of accumulation and subsequent neuroinvasion. We have, of course, done the converse experiments. If we take these mice at 56 days old, we then see accumulation of the prions as we'd expect in the ILFs in the small intestine, but we still don't see anything in the large intestine of these mice. So it suggests that the large intestine, for some reason, does not accumulate prions after oral exposure, and they don't spread from there. However, that doesn't mean that the large intestine gults, as has been shown in natural cases, can't accumulate prions. If we look much later on, so these are the control mice which eventually develop clinical disease, and then look in the colon, we then do start to see accumulation of prions in those large intestinal lymphoid tissues. So we think that the accumulation of prions in the large intestine occurs following the accumulation in the Peyer's patches. We think it's then spreading systemically via the bloodstream and lymph to other lymphoid tissues such as the spleen, the mesenchymal lymph nodes, and at the same time perhaps the large intestine gullet as well. And of course this is important because if people are using the appendices or the RAMLT to give some kind of gauge for the incidence of prion disease, they may be underestimated if they're looking at the wrong stage in disease, because if we'd looked in these mice up until about 4 or 5 months after oral exposure, we would not have seen this using a similar kind of tests. So I think this is important. Okay, so we've also been interested in co-infection. So all these studies so far have been in a nice healthy mouse in an SPF unit. But we also wondered, okay, so what would happen if there was some kind of pathology within the intestine? Would this influence the disease? Would it maybe exacerbate it, as Judy showed some really nice images of the parasites causing havoc into the gut epithelium? Does that make it more easy for the prions to spread across the gut epithelium? Or does the influx of macrophages allow to quicker dissemination, or maybe they destroy the prions more efficiently? And because David's project was focused on looking at the large intestine, we wanted an infection model which would be specifically restricted to the large intestine. And when I wrote this grant, the referees had their own opinions as to which parasite or infection model we should be using. They may have been correct or not, but I argued that we should use Trichuris murus, and the reason being in that it's a natural pathogen in mice. Importantly, it doesn't kill the mice, because I'm working on a very long disease of mice. There's some really nice infections we could have used, but the mouse is dead on its back within about 7 or 14 days. It's not much use for studying prion disease pathogenesis. But importantly, we don't need to treat the mice with antibiotics or use an attenuated parasite or virus, etc. So we can use our nice prion disease model of natural oral exposure and superimpose onto this the Trichoris murus and see what it has. And you can see this is just one of David's confocals, and fortunately our parasites are naturally autofluorescent. But this is B cell staining. You can see in our control mice we see the typical isolated lymphoid follicles, but this also causes exacerbation of the ILFs in the large intestine. So there's all sorts of interesting pathologies going on here which may influence prion disease. And so David did a rather heroic experiment of exposing mice to prion infection. This is the T. murus infection, and we exposed them at different points of the parasitemia, and unfortunately found absolutely no effect on the parasite incubation period. Yeah, I know, we were quite surprised and disappointed after a year and a half's work and got nowhere. But that is interesting because it informs our knowledge that, well, we don't see any accumulation of prions in the large intestine, and an infection which is restricted to the large intestine likewise does not exacerbate infection. Of course, now what we'd like to do is choose an infection which is specific to the small intestine and see what effect that would have. Of course, trying to find, again, for the reasons I've pointed out, a useful one is quite difficult, but I'm sure you guys would have a useful suggestion. However, it might skew the distribution of the prions in the body. So this is now, I think, looking at the cecal patches, and you can see, as we'd expect, we don't see much evidence of prion accumulation normally in the large intestine gullet. However, we did after exposure to T. murus. So maybe there's something interesting going on there. However, so David was obviously very despondent about that, but came to me one Friday afternoon with the typical Friday afternoon experiment, saying, well, how about we do this other experiment? And as a typical PI, I said, no, it's not worth doing it. I was wrong. David wondered, well, how about if we wait until the mice are about to show clinical signs and then hit them with a parasite infection? Maybe that might influence the pathogenesis. And this is interesting because there's anecdotal evidence that, for example, from old people's homes, that patients with Alzheimer's disease see an exacerbation of their clinical signs if there's some kind of chronic inflammation or if a bout of gastroenteritis or something goes throughout the home. I mean, it's anecdotal, but there's something to suggest that a chronic inflammation might influence what's an ongoing CNS disease. And David was also alerted to this particular paper here from Manchester, which they used a stroke model and overlaid T. murus onto that and showed that it actually exacerbated stroke if there was a T. murus infection ongoing. So David thought, okay, well, let's do the same with scraping. And a useful thing with working with T. murus is, as Julie pointed out, the dominance of the Th2 response when we use a high dose of worms, but we could actually also induce a Th1 dose by dropping the dose. So we could study the effects of a Th1 and Th2 immune response on CNS prion disease. And we use this particular model here. So we use an IC injection, so that the infection is much quicker. It takes about 170 days. And we timed the peak of the inflammation of the parasite infection to occur at either 84, 112, or 140 days after prion infection. Essentially, there's no overt signs of prion neuropathology here. The onset of prion disease neuropathology here, and these mice, in a few days' time, will start to show clinical signs. So we use these three particular time points for the high dose and the low dose to see what effect you had on prion disease pathogenesis. And fortunately, David found an interesting effect in that if we waited until the mice were just about to show clinical signs and then timed that parasite infection, we significantly shortened the incubation period by about 2 weeks, suggesting that a low-dose T. murus infection, which induces a Th1 response, could exacerbate the CNS pathogenesis of T. murus. But this was specific to the Th1 inducing dose. If we gave the Th2 inducing dose, we found no significant effect. So there's clearly something interesting going on there. Why is this occurring? Now, the stroke study that I alerted you to looked at that study suggested that the chemokine CXCL5 was important, and that if they block CXCL5, they could block the effects on stroke. And during scraping infection, we see an elevation of CXCL5, or otherwise known as RANTES.
which is reasonably similar to what this study found following T. muris, and we found exactly the same. But this was not specific to a low or high dose of T. muris. So we don't think that the effects we're seeing have anything to do with CLCL5, and this is mirrored in an independent study looking at CCR5 deficient mice. They've given them prion disease. There was no effect of this deficiency on prion disease. So we don't think that the effect we're seeing on prion disease has anything to do with CLCL5 or RANTES. And David's trying to follow this up now to study some other candidate molecules which may be causing this effect. So hopefully in the time I was allocated, I've been able to convey to you how we think that prions are hijacking the GALT to then spread into the peripheral nervous system, which then they go on to and cause CNS disease. I'd like to thank, of course, all the work that I presented was done by a talented team of students and postdocs in my group, and Anuj has a slide on, has a poster at the back on some of the MCell work we're doing. Like to thank the colleagues in Team MCell, which were important, and likewise parasitology from Catherine Else. Thank you.